Okay, for this video, I just wanted to do a short recap on the synapse and a little bit about neurotransmitters. We've already covered this pretty well in the Chapter 11 material, so hopefully this is just a reminder. Remember, there are three parts to the synapse. We have the ending of the axon, which is known as the presynaptic axon terminal. Then you're going to have the postsynaptic membrane and the space or gap in between called the synaptic cleft. So the presynaptic axon terminal is ending, that is sending some message via a action potential that hits the end and then causes neurotransmitter to be released. The postsynaptic membrane is going to have receptors for that neurotransmitter so that the message is received, and the gap in between the two is the synaptic cleft. You throw these three things together and those are called a synapse. So as it turns out, as we're going to see, especially when we get to the brain chapter, that a neuron can have a tremendous number of synapses. We've been looking at very simplistic approaches in terms of one neuron, one synapse, but on the average, a spinal motor neuron in the CNS can have an average of 10,000 synaptic terminals or knobs from other neurons. And on average, that's the breakdown. About 80% will end up synapsing on the dendrites, about 20% on the cell body itself. When we get to the cerebellum, we're talking about neurons with 100,000 synapses. And you might start to think, well, how do we ever make any decisions if we have like 100,000 screaming kids yelling at us? How do we ever make a decision based on what they're screaming at us? And the brain has a way. We'll talk about that in the last video of this chapter. All right, so we have the axon terminal, contains the synaptic vesicles which possess neurotransmitters. We've already discussed those in terms of acetylcholine. These are chemical messengers, and they'll diffuse across the synaptic cleft. Typically can be broken down by enzymes. It can also be diffused out of the synapse as well. There are different ways that they can be broken down. They can also be what's called reuptaked, so they can actually travel back to the presynaptic terminal and end up being taken back up by that presynaptic terminal. So here is a cell body. You can see the cell body in here, all of these different synaptic terminals, presynaptic terminals, or knobs if you wish, acting on that cell. It's just an incredible array. And here are the vesicles filled with neurotransmitters. This is the presynaptic membrane, the synaptic cleft in between, and then the postsynaptic membrane where all the receptors are. And you can see how the vesicles are going to congregate in a certain area, and that area just happens to be across from where all the receptors are located on the postsynaptic membrane. This is the highest density. So the vesicles will tend to be transported or chaperoned right into this region of the presynaptic terminal. And you've seen this picture before. Neurotransmission, just to refresh our minds, is a action potential arriving at the presynaptic axon terminal. The voltage opens voltage-gated calcium channels. The calcium floods in. Calcium activates proteins in the presynaptic terminal that will usher the vesicles to the membrane of the presynaptic terminal. They'll fuse, and in the process of exocytosis, the fusion will allow diffusion of neurotransmitter into the synaptic cleft. It will travel across the cleft, bind to the receptors on the postsynaptic neuron. In the case of acetylcholine, which this is modeling, it will open those receptors and allow sodium to enter. This will induce another action potential on the membrane of this cell. There are two classes of neurotransmitters. There are those neurotransmitters that cause an excitation or the possible development of an action potential, and then there are those neurotransmitters that make it less likely to occur. And so excitatories promote action potentials versus the inhibitories that will typically suppress action potentials. So we'll get into that more here in this next video. Okay, so Why does a neurotransmitter have the effect it does? Well, remember, it's all about what the receptor is about. There are multiple types of a receptor for a neurotransmitter, such as acetylcholine. There are two major different types of acetylcholine receptors, one called muscarinic, one called nicotinic, and they have completely different actions on a cell. So you'll see that it's not only the neurotransmitter, but it's the type of receptor that that neurotransmitter binds to. For serotonin, there are currently 19 different types of receptors for serotonin that have been classified. So it's not such a straight and simple picture if you just say serotonin and its receptor or acetylcholine and its receptors. You have to be more specific. All right. Okay, so it can promote action potentials, but it inhibits cardiac neuromuscular junctions, and because those are two different types of receptors involved. Okay, so just to review, just to remind you from what happened in the muscles, cholinergic, when we say it's a cholinergic synapse, that means acetylcholine is involved. Okay? All neuromuscular junctions, as we mentioned before, utilize acetylcholine, as do several synapses in the central nervous system. We'll talk about that in more detail here in a bit. Okay, turns out that all synapses in the peripheral nervous system that are neuron to neuron utilize acetylcholine. You'll see that when we get to A&P2, the first chapter. And then all glands function due to actions caused by acetylcholine. What happens? Remember, just to review the steps, action potential arrives, then we have the depolarization, which allows calcium to enter. This causes fusion of the vesicles and exocytosis. Then, once the vesicles fuse, it will cause exocytosis of acetylcholine. Acetylcholine will bind to its receptors and depolarize the postsynaptic membrane, causing a new action potential to develop. The breakdown involves an enzyme called acetylcholinesterase that cleaves the acetyl group from the choline group. And recall that choline will be taken back up by the presynaptic terminal. So let's look at these pictures that show you this, and once again delineate the steps. I invite you to look at the PowerPoint for this chapter that I give to my face-to-face crowd, and it will give you a bit more time to look at these pictures. Though you've seen these effects before in chapter 11, it never hurts to review again. All right, see you next time.